Hello, my name is Rinki Ratnapriya and I'm an assistant professor at Baylor College of Medicine. In today's tutorial, I will provide an overview of genomic methods used to connect genome-wide association findings to biological functions. We will focus on how these methods are applied for identifying causal variant, genes, and underlying mechanisms in age-related macular degeneration. The genetic basis of common disease is a subject of significant interest and an active area of human genetic research in post-genome era. Genome-wide association studies, or GWAS, has been instrumental in bringing us closer to this goal faster than ever before. So what is a GWAS? GWAS is a genetic mapping approach that examines the DNA of a large group of people to identify genetic markers that are associated with a disease or trait. GWAS studies look for small variations in DNA called single nucleotide polymorphisms or SNPs that occur more frequently in people with disease or trait compared to the controls. A typical GWAS can include millions of common SNPs. Shown here is the schematic of a typical GWAS approach, where you will collect a bunch of cases and controls, and then you will genotype millions of SNPs that are spanning to all the chromosome in the human genome to identify the variants which are occurring at higher frequency than in control. Age-related macular degeneration, or AMD, is the leading cause of visual impairment in elderly population that has a strong genetic component. Thus, AMD research has been at the forefront of this movement, primarily GWAS of AMD, that has achieved rare and unprecedented success among complex traits. The first ever GWAS was performed for AMD in 2005 with merely 96 cases and 50 control that identified the CFH locus. In 2016, that number was increased to 16,000 cases and 17,000 controls that identified 34 associated loci. And the most recently, in 2024, 61,000 cases and 364,000 controls has led to the identification of 63 associated loci for AMD. However, the interpretation of GWAS finding is not always straightforward. Let's take the example of this locus pilare for AMD. What you are looking here is the locus zoom. X-axis, it shows the genomic location, and the y-axis shows the log 10 p-value. So for GWAS, since we are testing millions of SNPs, genome-wide threshold needs to be applied, which comes down to 5 times 10 to the minus 8. So the variant with the lowest p-value is generally assigned as the risk variant. So in this case, this diamond-shaped SNP, which we also call the sentinel SNP, is associated with AMD. Similarly, the gene closest to the variant is given the locus name. For this example, pilare is right here, and that's why this locus will be called pilare. However, you can see that there are many more variants and genes in this region that are crossing. So for these variants, they are also in high LD, which is linkage disequilibrium. That is why it becomes very, very hard to say that which is actually the causal variant, and any of these variants that are in high LD and are associated with AMD can be a candidate. Similarly, for this region, pilare is the closest gene, but that does not mean that that is the only gene responsible for the disease. So that is why the functional validation is needed to identify the causal variant and the causal gene. Additionally, majority of the GWAS variants are located in the non-coding region of the genome. That is why it is hard to interpret their functional meaning. So what are the causal variants? Whether these are the common variants, or in addition there are rare variants that can be important for AMD. Secondly, we don't always know the disease-relevant cell types by the genetic finding, whether the regulation is cis or trans, and whether those regulations are cell type specific. And finally, connecting with the known biology is difficult, as we don't know the target gene, and we don't also know that how these risk genes interact with each other and what are the pathways involved. Although the majority of the variants associated with AMD are common, The role of the rare variants with the minor allele frequency less than 0.1% have also been investigated by imputation using disease-specific custom arrays and the whole exome and the genome sequencing. Rare variants play unique roles in the genetics of AMD because of their distinct feature. In contrast to the common variant, they offer hypothesis-free evidence for the gene causality, a precise target for the functional analysis for the better understanding of disease mechanism, a new favorable target for the drug development, as well as a genetic marker with the high disease risk for personalized medicine. Targeted exome and family studies have identified rare variants in CFH, C3, C9, and CFI, establishing these genes as the causal genes for AMD. In a recent study, we analyzed 263 individuals from 63 families and identified several candidates, including SPEF2 and BCAR1, that are the newer genes that have not been implicated in AMD before. However, a better estimation of the functional impact of rare variants across whole genome is needed to enhance their contribution to improvement in human health. So this slide summarizes the genetic architecture of AMD as we understand from the GWAS and the sequencing studies. We have few common alleles and the rare alleles with the high effect, for example, CFH, CFI, C3, and the CFH and the ARMS2 locus. However, majority of the alleles or the variants associated with AMD, they are common with moderate to low effect. As majority of the AMD genetics is explained by the common non-coding variants, how do we comprehend their function? So in 2010, Matthew Morano showed that the GWAS variants, they often overlap with enhancers, promoters, and the open chromatin regions. Thus, risk variants are likely to exert their effect through gene expression regulation. We know that the gene expression and their regulation can be very tissue and the cell type specific. Thus, studying the gene expression and their regulation in the disease-relevant tissue is the first critical step and a prominent tool for biological interpretation of the GWAS findings. So this slide describes the commonly used omic tools that are used to connect the genetic variant with the gene expression regulation. So first is the genome level, where we need to find all the genetic variants. That can be done either by SNP genotyping or the whole exome and the genome targeted sequencing. Second, we need to find the transcriptome, which can be achieved by RNA sequencing and the single cell sequencing, and the epigenome that can be done by ChIP-seq, ATAC-seq, or the DNA methylation. So a genetic variant can impact many different molecular phenotypes, including expression. In that case, we call it EQTLs, or splicing, which is SQTLs, protein abundance, which is protein EQTLs, and methylation, chromatin accessibility, histone modification, or any other kind of molecular phenotype. So for this tutorial, we will focus on expression EQTLs and the methylation EQTLs, which have been already published in the context of AMD. So here it is shown that how the EQTL works. So suppose there is a SNP which is near to two genes, gene A and gene B. And if you measure the gene expression in a large number of individuals with varying genotype, so suppose the genotype could be AA, AT, and TT, and you can see that the expression of the gene A is very changing with respect to the genotype, whereas the expression of the gene B remains the same. So you would conclude that this variant is acting as an EQTL for gene A and not for gene B. EQTLs can be of two different kinds, cis EQTLs and the trans EQTLs. In cis EQTLs, the sequence variation that controls the expression level of the transcript lies within or in close proximity to the gene, usually 1 MB from the transcription start site on either side. Whereas in the trans EQTL, the genetic variant that affect the expression of a gene can be located far off, more than 1 MB, or even on a different chromosome. Shown here is the typical pipeline for identifying the EQTLs. It usually begins with the tissue collection, usually for the disease-relevant tissue, which would be retina, RPE, and choroid for AMD. You would isolate the RNA, perform the RNA sequencing, followed up by normalization, batch correction, covariant, and generating a gene expression profile for all the individuals in the study. From the DNA side, you will be isolating the DNA, performing the genotyping either by SNP array or genetic sequencing, followed by the imputation, and then once you have both the RNA sequencing and the genotyping data available, you can apply different tools, for example, matrix EQTLs or the QT tools, and then you will find out the cis EQTL for each and every variant and then perform statistical analysis to find out the variants that are at the genome level regulating the gene expression. So this relationship between the gene and the variant is called the cis EQTL, where the variant is called the E variant and the gene is called the E gene. In 2019, we published the transcriptome and genetic analysis of 500 donor retina to generate much needed reference of EQTLs. In this analysis, we identified 18,000 genes to be expressed in retina, and of these 10,474 genes were regulated by 14,565 variants, which means that sometime one gene was regulated by more than one variant. Now we will review how we will use this EQTL data to understand their role in overall complex trait, AMD in this example. Then how do we use this data to find the causal variant and the target gene for the known AMD loci, and how do we search for the novel association? So first we are interested in identifying whether the EQTLs that were identified in retina are enriched for the GWAS signals. So to do that, you would take the AMD GWAS data and then you would ask the question whether the variants that are associated with AMD are also in EQTLs. We also did it for the other different kind of complex diseases, and we found that retinal EQTLs were enriched for the GWAS signals, and the shared EQTLs enrichment is significantly higher than the tissue-specific EQTLs, which has also been reported for other diseases. So this data can be used to find the causal variant, what we call as the co-localization studies. This method determines whether a single variant is responsible for both GWAS and the molecular QTL signals in this locus. So the co-localization methods seek to estimate the probability that the complex trait and the expression causal variant are the same. Shown here are the results of the co-localization studies. Using AMD GWAS data and retina EQTL, we were able to identify the target gene and the causal variant at 3 different loci. For example, at B3GALTL locus, B3GLCT gene was regulated by the SNP, where you can clearly see the gene expression changes in response to the genotype. Similarly, at RDH5CD63 locus, block C1S1 was identified as the target gene, as well as the ACAD10 locus SH2B3 was identified as the target gene. So there have been few studies done on the retina and the RPE QTL, and if you combine all the data, there are few themes that emerges. First, within a single locus, more than one SNP can be an EQTL, which can regulate single or multiple target gene. For example, if you look at the CFI locus, you can see there are 2 distinct variants that are regulating the same gene. Secondly, the lead SNP is not always an EQTL, suggesting other variant within associated loci can regulate the gene expression and thus can be causal. Finally, one locus can have multiple target genes, and that too in different tissues. For example, for AMD, both retina and RPE has significant role, and you can see at the RDH locus we identified block C1S1 at the retina side as the target gene, whereas on the RPE side RDH5 seemed to be the target gene. EQTL data combined with the GWAS data can also be used to identify novel association using what we call as transcriptome-wide association analysis, which aims to identify gene associated with a particular trait, AMD in this case, by examining how genetic variation influences the gene expression levels. So using this data, we identified 23 genes at 16 loci outside the known GWAS association, and that represent the strongest new candidate for AMD-associated genes. EQTL studies can have certain limitations. First, the multiple candidate genes can be identified within a single locus, making its interpretation difficult, requiring functional validation. Secondly, most fine mapping statistical approaches don't consider multiple causal variant within the same locus. However, with the recent literature, we know that within the single locus, multiple causal variant as well as multiple causal genes are possible that can be influencing the phenotype. Also, the bulk tissue data, EQTL data that are most widely available, they lack the cell type specificity. However, these EQTL studies that has to be done at the population level require certain sample size. For example, for EQTL, a sample size of 80 is minimum required. Finally, the EQTLs are severely underpowered for any kind of trans-EQTL analysis, which can be contributing significantly to connecting the GWAS to the function. Similar to expression level, we can also use other molecular EQTL. Shown here is the example of the QTL mapping of the human retina DNA methylation, where the DNA methylation profile of 160 human retinas with the co-measured RNA-seq and the 8 million genetic SNPs were used. It uncovered the sites of the genetic regulation in the cis 13,747 DNA methylation loci affecting gene expression, with over 1/3 specific to the retina. Finally, the summary database Mendelian randomization and the co-localization analysis identified 87 target genes likely mediate the genotype effect on the AMD. As most GWAS variants are located within the non-coding genome, SNP enrichment using chromatin annotation can be used to identify disease-relevant cell type. These enrichment methods integrate GWAS results with different genomic annotation and prioritize the cell type in which associated variant overlap annotation more frequently than expected by chance. So the open chromatin profiles can be used to estimate their polygenic contribution to heritability in AMD GWAS data. Here is a recent study which utilized ATAC-seq data in the single cell level in the human retina and RPE to identify the disease-relevant cell type. By analyzing this data, they identify RPE and glial cells as AMD relevant, and the fine mapping using the peak to gene analysis across all cell types in the integrated single nuclei seq and SN ATAC-seq data identified 22 genes correlated with 7 AMD risk loci. Other method that can be used for integrating the ATAC sequencing with the GWAS data are Garfield, LDSC, and Greger. So we briefly talked about the trans-EQTLs, where the variant is regulating the gene which is far off or even on a different chromosome. We know that our genome is 3D. So high-resolution genome topology methods such as 3C can be used to generate chromatin contact maps at high resolution. Shown here is the example where the retina maps were built using human donor eye, and combine integrating this data with super enhancer, histone marks, CTCF binding, and the other transcription factor, they were able to find topological associated domain stats which were regulating several retinal genes. And combining this data with the GWAS has a potential to identify the associated variant that are regulating a gene that are far off. So molecular EQTLs using expression data, ATAC sequencing data, or the epigenome data can identify a candidate target gene. However, that needs to be functionally validated with a putative regulatory variant. That can be achieved with several different methods. One is the cell culture based assays, which could be either single variant assays such as the luciferase assay, where you would place a cis regulatory element under the minimal promoter driving the luciferase gene, and you would compare the gene expression driven by the CRE and assess whether the variant is impacting the gene expression. The highly parallelized version of that is called MPRA, or Massively Parallel Reporter Assay, where you could test several hundred thousands of cis regulatory elements of the CREs. Similarly, you could use CRISPR tools to delete or modify the CRE to infer and confirm the effect of the CRE on the gene expression. Secondly, the animal models can be used, although AMD is a macular degenerative disease and the mouse does not have macula. However, the certain endophenotypes of the disease can be very well mimicked. For example, animal model can be put on a high-fat diet, or you can build the humanized model of the CFH, and there are wonderful examples of these in literature where they have been successfully used to find the certain phenotypes of the AMD. Finally, we can also use the stem cell model, which are going to be very popular in coming years. There have been already several studies where they have used iPSC-RPE and retinal organoid. For example, Smith et al. In 2019, they performed this iPSC-RPE genetic analysis, and they did the fine mapping of a AMD associated locus, where they were able to identify causal variant in the gene at the VEGF-A locus. So to conclude, here we have learned the integration of the functional genomic approaches using the transcriptome and the epigenome data, and the GWAS can identify causal variant genes and the cell type relevant to the disease. Heritability of the AMD can be significantly explained by the molecular QTLs, and these integrative approaches can be leveraged to identify new disease candidate. And finally, the functional validation should be performed in the disease relevant system to understand the disease mechanism. So to summarize, we begin with the GWAS data, where we have the genetic variant associated with the phenotype. When combined with different kind of functional genomic approaches at the genetic regulation, single cell, epigenome, or the 3D genome, we are able to build many different kind of molecular QTLs that can provide insights into the function of a non-coding variant identified in the GWAS study. Finally, we need to functionally validate using the cell culture model, IPS model, or the animal model to gain more insights into the disease mechanism of AMD. So what should be the next step? We have seen in this tutorial that molecular QTL analysis can be a powerful tool for connecting the function of a non-coding variant to the disease. However, many different kind of molecular QTLs have not been performed in AMD relevant tissues, as well as wherever the studies have been done, they remain underpowered. So first and foremost, a well-powered studies with many different kind of molecular QTL would be desirable. Secondly, most of our analysis has been done using the control data or by combining control and the AMD data. Thus, a comparative QTL analysis between controls and the AMD could reveal unique insights into the disease pathology. Thirdly, most of the data are bulk. What that means is that we lack the cell type resolution for these regulatory information. Single cell remains expensive. However, in coming years, technology will get cheaper and cheaper, and thus the single cell resolution EQTL analysis will provide insights into the cell type specific regulation, which is much desirable for any disease, including AMD. Finally, integrating additional modalities such as 3D genome as well as spatial transcriptomic can add to unique insights into the disease. And finally, high throughput functional validation. That remains a key bottleneck right now because all these molecular QTL analysis can be done high throughput way. However, most of the functional validation is done by one gene at a time. In recent years, iPSC-RPE model and the organoid model have become much more feasible, and it is possible that in coming years we'll see generation of high throughput models using these organoid and iPSC-RPE for the functional validation of the AMD GWAS. Thank you for your attention.